Now that we have some idea of the principles on which molecular motors operate, let's take a look at some specific examples. The electron microscope image here is of sarcomers in cardiac muscle cells. The model for these banded structures is shown below. They're composed of actin filaments, and we introduced actin as an example of a structural protein right at the beginning of this section, interspersed with other filaments on which sit myosin molecules. When the muscle tissue is activated, these filaments are pulled in between the actin cross bridges, and we'll take a look at how that occurs. Myosin has a dual-headed structure, so here are some electron micrograph images of the myosin heads, which have this dual-headed structure, and this long filament here that attaches to the central filament that gets pulled into the actin units. So this is not drawn well to scale here in the sense that the two heads that grasp the actin are quite small and the connection itself is quite long. The heads are where the action is. Myosin is not a rotary motor, rather it's a walking motor. This has been worked out to a certain degree using single molecule optical fluorescence techniques in which the motion of myosin heads along an actin filament could be recorded with a high degree of precision. It turns out that the myosin really does perform a walking motion in which one foot will unbind, swing ahead of the first foot to cause an advance of one step on the actin template. At the time of the recording of this particular module, I've been privileged to see a beautiful atomic force microscope set of images taken with a very high speed AFM, in which this motion of myosin heads on actin filaments has been recorded with molecular resolution, and the binding and unbinding of each of the heads as a function of ATP and ADP concentrations measured. Here's a perhaps even more remarkable motor, ATP synthase. This motor can perform two functions. As a motor, it's driven by a proton gradient across a membrane. So this part of the motor here sits inside a lipid bilayer, and there is a shaft here which allows this head to rotate. Proton translocation causes rotation of the head, and simultaneously, on binding of ADP on this side of the membrane, the incorporation of phosphate ions. So this motor, under the action of a proton gradient then, is what synthesizes ATP. So a proton gradient is the energy source then to drive the reaction ADP plus P goes to ATP. The P standing for phosphate here, so please ignore my chemical simplifications. The same type of motor structure can hydrolyze ATP to turn the motor the opposite way and pump a proton gradient. So this is a remarkably useful structural motif in biology. Rotary motors somewhat like this are found in swimming bugs that move by rotating flagella using a motor like this at one end of the bug. So this shows the two functions of the motor schematically. In this case, a high concentration of protons on this side of the membrane is driving the reaction between ADP and phosphate to form ATP. In this case here, ATP, when present in high concentration on this side of the membrane, is being hydrolyzed to ADP, driving an increase in proton concentration on this side of the membrane. Here is a beautiful movie showing what can be done with single molecule optical techniques. In this movie from the Kinosita lab in Japan, the F1 unit of ATP synthase, which is the basic rotary module, was attached to a glass slide. Recombinant biochemistry techniques were used to then glue a large actin filament to the top of the motor, and the actin filament was loaded with dyes. So although the motor is too small to be seen in an optical microscope, the actin filament is resolved as this anisotropic blob here, and you can see how when the motor is supplied with ATP, the actin filament rotates fairly systematically in 120-degree steps, although you notice every now and then there is an attempt at a back step. This is, after all, a fluctuation-driven system.